Good morning, everybody. This is Mr. Zimber coming at you. Today we're talking about enzymes. This is part one of two parts on activation, regulation, and saturation. Um, and we're going to be talking about how enzymes, particularly today, talking about how enzymes are activated and what factors influence their reaction rate. So what you should do to get started today is go ahead, title this Enzymes, Activation, Regulation, Saturation in your notes, part one. Write down the essential question here. And then there's also an accompanying worksheet that I will have for you at the front of the room that has 3 different graphs on it. So go ahead, get your notebook set up, take some time to do that right now, and then when you're done, you can unpause the video and continue along. So do that now, and then you can click play again. Okay, a reminder about enzymes. We have covered this already, but just so we're all clear here, an enzymatic reaction happens like this. We have the enzyme denoted up here by E interacting with the substrate denoted here by S. E plus S goes to the enzyme substrate complex, which will then undergo a reaction. The substrate will undergo a reaction, and the product or products will get released. The whole process here is an enzymatic reaction, and the rate at which new products are formed is the reaction rate. Okay, so we know substrate binds at the active site to the enzyme, a reaction occurs, or the products get released. That is our enzymatic reaction, and today we're going to be talking a lot about the reaction rate. So when we're saying reaction rate, we're saying how many times is this happening in a second or in a minute or whatever, okay? So keeping that in mind, let's look at these two graphs. And what we're going to do here is these two graphs, figure 1 and figure 2, are in your note sheet, which should be pasted into the left-hand side of your notes today. And what I'm going to have you do is reflect on these two graphs before we continue. So I want you to think about what they are saying and what they mean. Figure 1, we have temperature versus percent activity of the enzyme. Figure 2, we have pH, which is to say acidity or basicity, and its activity of different enzymes. This one has a single, figure 1 has a single enzyme. Figure 2 has 3 different enzymes with graphs and how their percent activity changes over those, either over temperature or over the pH. In your worksheet, it's going to ask you about the manipulated and responding variable. Remember, the manipulated one is what we're purposefully changing, and responding is what we're measuring. And then it's also going to ask you to answer these questions here. Where does this temperature peak? And here, where might you expect to find each of these enzymes given their preferred pH level? Okay, go ahead. That's graphs 1 and 2 in your worksheet. I want you to answer the questions in your worksheet before we continue to start thinking about this and what they mean, what these graphs are telling us before we continue. Okay, so please do that, and then when you're ready, you can click play again. So pause now, then click play. Okay, here is the first graph. And what this graph is telling us is 1, the reaction rate of an enzyme is dependent on the temperature. And what do we mean by that? Well, this percent activity means how active is the enzyme. Higher activity means the reaction rate is going to be higher. Okay, so at 0 degrees, the enzyme has maybe 5% activity. At 40 degrees Celsius, it has almost 100% activity. And then it drops off precipitously after 40 degrees. And if we track this curve here, we see that an increase in temperature is leading to an increase in activity. And part of this has to do with this thing, or the idea of kinetic energy. We've got molecules that are bouncing around, and the hotter the solution gets, or the more heat it has, the more kinetic energy it has, the faster the molecules are running into each other and also running into the enzyme, which means we have a higher chance of the enzyme bumping into the substrate and that reaction proceeding to the product. Okay? Yet, it doesn't go up forever. Once we get beyond 40 degrees, it drops off really quick. Why might this be? All right, drops off really quick. In other words, the hotter it gets, the faster the molecules continue to move around. You would maybe predict that it just keeps going up and up and up and up. Instead, it doesn't. And why is that? Well, it's because of something that we call denaturation or a denatured enzyme. This is one of the key ideas from this lesson that I want to make sure you put into your notes. Once an enzyme gets too hot, too high of a temperature is reached, the hydrogen bonds that give it its tertiary structure, its three-dimensional shape, right? So let's say this is our original amino acid sequence, then hydrogen bonds form between that amino acid, and it turns into this folded chain, which I'm depicting with my hands. If it gets too hot, that chain is actually going to start to unravel again and turn into its main structure, which was just a regular flat chain. When that happens, when it loses its structure, it loses its function. We call that denaturation. A denatured enzyme has fallen apart into its primary and secondary structure and is no longer able to function. All right? Which means that, remember, the enzyme is catalyzing this reaction, boom, it starts to denature. It goes from functioning to not functioning. And when that happens, it can no longer catalyze the reaction. So its activity plummets. So at around 40, 50 degrees, this enzyme is going to denature and lose its ability to function. Temperature affects enzymatic reactions on a slope like this. Okay, so here we've got an example. We've got egg. It's made out of proteins. In this case, we're also showing modeling what a protein would look like. So here we have a real protein in the egg, and here we have fake protein that we're modeling with paper clips. Before denaturation, the paper clips they're folded up and they are functional. They can still do a very good job holding paper together. All right, they maintain their paper clip shape. They can still clip paper together. Similar to this egg here, it has functional proteins in it that would serve to help this little chicken grow if that were to be fertilized. However, we're talking about this temperature range right here, between 0 and 40 degrees. We know that when we cook an egg, it turns white. And the process of going from clear to white is a demonstration of a denatured protein. So this egg is getting denatured when it gets cooked. Similar in our model down here, these paper clips, if we were to heat them up, they would denature, they would lose their shape, and no longer be able to hold paper together. They would lose their function. Let's look at that. Okay, so again, this denatured protein is what we see happening at this part of our graph. Okay, it gets denatured, it loses its shape, and it loses its function. So the activity of our protein or our enzyme is going to decrease all the way down to 0 because it's completely non-functional. That's just an example there. Okay, part 2. This is the second graph that you looked at. Hopefully you filled out the worksheet for this one as well. And what we learned from this is that pH can also affect enzymatic reactions. The pH it's a measure of how many basically how many hydrogen ions are in the solution, how acidic or basic a solution is. And pH can alter the structure of an enzyme also by interfering with hydrogen bonds, which will also lead to denaturation. It can also do other things that affects the functionality of a protein, and therefore the functionality of an enzyme. Here we have 3 different enzymes that peak at various pH levels. All right, so we see the blue one here is peaking at a very low pH, which means high acidity. The yellow one is peaking at around 7, which is neutral. And then the green one is peaking at around let's say 11 pH, which is very basic, okay? And again, we see as it approaches this ideal pH level, it's at its highest functionality, highest percent of activity, and if it goes in either direction, it falls off in its activity. So these enzymes are very good at doing their jobs in specific pH environments. This one at a very acidic, this one in neutral, and this one in high basic environment. So where might we find these? Well, a low pH means it's an acidic environment. So this blue one is going to be found in some sort of acidic environment, such as the stomach. It's a very acidic place. Pepsin is one such enzyme that helps in our digestive system. So we might think that blue could be a stomach, something, an enzyme you might find in your stomach. This yellow is a neutral pH. This enzyme functions best in neutral environments, and water is a neutral, or I should say the water in our body is neutral. It's at a pH of about 7.4 typically. Most enzymes in our cells are going to function best in this area, around 7. And then a high pH means it's basic. This enzyme might be found in monolake, which is a very acidic, or I should say alkaline environment. And it was previously thought to be uninhabitable, but scientists are now finding that there are bacteria that can survive in this really alkaline lake. I have it linked here if you want to read more about that particular lake. But we could imagine that if we found an enzyme that functions best in a high basic environment, possibly it's an enzyme that came from one of these bacteria, okay? Because the enzyme needs to be able to function in that really basic environment. Okay, and then here is our third graph today, and similar to the other ones, I want you to answer in your worksheet. This is the third graph. I want you to answer or figure out the manipulated variable. What are we purposefully changing? Responding variable, what are we measuring? And then I want you to answer that analysis question. In this case, what is the relationship between substrate concentration and the reaction rate? Okay. On the x-axis here, we have the concentration. Y-axis, we have the reaction rate. Go ahead, make your notes for that final graph today. And then when you're done with that, we will continue talking about this graph. Okay, so go ahead and do that now. Pause the video and then come back when you're done. Okay, if you're back with me, presumably you have done the assigned duty of filling out the worksheet. And what this is showing us is something that's called the Michaelis-Menten model of enzyme activity. And what that model says, the Michaelis-Menten, is that the rate of an enzymatic reaction is dependent on the concentration of the substrate. In other words, if there's no substrate, obviously there's zero reactions happening. As you increase the substrate, it's going to go up very quickly, but at a certain point it's going to start to flatten out. The reaction rate is going to start to flatten out because there is a limit to how quickly any specific enzyme can actually do the reaction. So if you think about it, in terms of the toothpicks that we did yesterday, you had a whole pile of toothpicks and they're all right there available to you, but you could only break them so fast, right? It didn't matter. You didn't have to go searching for them or anything. They were right there for you, but your hands were only so quick. There's a limit to how fast you could break them, and that's what this graph is telling us. Okay, so each enzyme will have this thing called a Vmax, which is its operating or its peak operating rate, essentially. It's the fastest reaction rate it can sustain. And then when it approaches Vmax, that means that it's saturated by these substrate. It doesn't have to search for them at all. It's just right there. And so yeah, the Michaelis-Menten model tells us that there's a limit to how fast an enzyme can do any particular reaction. It's dependent on substrate concentration, and that is on a log scale. It increases at a log scale. All right, here's the summary that I wrote up for this article. or this lesson. What I want you to do is write up your own in your notes today. So you should have on your left page the 3 graphs that you have identified the manipulative responding variables, and you have identified or answered the questions in that worksheet. You should also have notes from today's lesson regarding the effect of temperature, pH, and substrate concentration on enzyme reaction rates. Okay? When you're done with that, you're going to write up a summary, and then you'll be done for today. So go ahead, write your summary. Thanks for listening today, and have a great rest of your day.